A few weeks later, Chinese scientists had sequenced the first COVID-19 genome and released it throughout the world. And that people started to do development of vaccine and many things going on from there, including tracking the COVID virus, and also how do we know about the mutation, also the silent mutations? That's because of sequencing data. And that's why I want to start how important about sequencing. Okay, let's go through the more detail about my talk today. You already know, many of you that know about the epidemiology, which is the science that study the pattern, causes, and effect of health disease condition in the defined population, right? So, but the 4 basic questions in epidemiology that when you study on this topic, which is what it is, what is it that causing the diseases? Has it seen before, or is something that completely new, or is something that we have seen previously? How can we fight it? And is it an outbreak? Is it part of this outbreak, or is not relevant at all? So this is like a basic question in epidemiology, and this is what we normally do. We normally do identification to know the species of the bacteria, for example. And if you want to do anything below species level, and that we call typing, for example, we want to know the serotype, we want to know the MLST type, even we want to know the resistant genes, that we call typing. And all of these things, we normally do by this step, right, in the lab, by getting the sample, do media culture, gram straining to know the gram positive or negative, multi-talk to do the species. And further on, my point is, one answer that you want from the lab, you need one specific experiment. If you want to know 10 things, you have to conduct 10 different experiments. And that would cost weeks to months until you get all the answer you want. But since if you have sequencing data, many of things that you see here will be replaced by the sequencing data. Once you get the culture, you do the pure culture, you put your culture strand into sequencing machine. And after you getting sequencing data, you can know species, you can know how the strand related, how you know if it contain any resistant genes, you know if it contain any other virulent gene, you know all of these things just by looking at the sequencing data. You just look at sequencing data just like information. Every information about the bacteria or even virus, they are already in the sequencing data. But the problem is, how can you crack that information out of the sequencing data, which I will show you later on. Okay, this is an example of I think many of you have already know this format or this type of data we call FASTA format. The FASTA format is a standard format that you normally store nucleotide sequences or amino acid sequences, DNA or protein sequences. In this FASTA format, you normally have 2 components, the ID and the sequences. But this is not the data that's coming out right away from the sequencing machine. When you sequence it, when you do sequence whole genome of bacteria or virus, you normally chop them into smaller pieces of DNA from the whole long or circular chromosome. You chop them into small pieces of DNA we call reads. And the data of these small pieces of DNA that you actually put it in sequencing machine. Of course, before you put it in sequencing machine, you have to do some kind of library preparation, put adapter, put many things in there for the machine, for the sequencing machine to be able to recognize and start sequencing. And this is the data that's coming out right away from sequencing machine. We don't call them FASTA anymore. Now we're gonna call them Fastq because it contains FASTA and quality score. So this part, the first 2 lines, it looks like the FASTA format that you have seen before. But the last line is the sequencing quality. Every base pair that got sequenced, it has quality. And of course, you don't understand how good or bad the quality is meant for the computer program to understand. But my point is, this is the actual data that you normally get from sequencing machine, the Fastq format. And what you can do with this data, you can assemble them from the small reads, small sequences. You can try to assemble them to something that big enough for you to identify any genes or even complete them to the circular chromosome of the species that you study. This is called assembly process. And another thing that you can do, you just map these small reads to a reference genome. You might want to identify any differences between your strand and the reference genome. And you also can do by the mapping. All of these things, you will not do it by yourself, but you use computer program to do it. And if you ask me about how can you find this sequencing data if you don't have one, you can actually, those sequencing data, they're all publicly available from the previous study that many scientists had already studied. They are in these 3 main databases and they are all synchronized. So in the DBJ in Asia, NCBI, of course, in the US, and EBI in Europe. Okay, that's very short, briefly about the sequencing data. And now I'm gonna get you through how can we actually apply or using those sequencing data for typing and surveillance of infectious diseases. Okay, of course, now you have the data, but the data is just, you can see the sequencing data just like languages. You need some kind of interpreter to understand that languages. And the interpreters that I'm talking about to understand the sequencing data is called bioinformatic tools. And these bioinformatic tools that make sense of the sequencing data ATCG, most of them, they are in the platform like Unix, which you have to use a command line, Unix, Linux command line in order to install and running the program, which mainly for those who have advanced computer skill to be able to use it. But only a small number of the tools, they are with the user interface, which you can use by using your Windows machine or Mac computer. But those are commercial tools, which are quite expensive. So to overcome this problem, our research center also put many available tools and make it with the user interface in the form of web-based tool. I want to disclaim here, actually, what I'm going to show you, there are other tools that can do the similar things. I just want to show what we have because of the time limit that I have. And I want to show you some example that we have to show you how to use it, the bioinformatics tool to do some kind of typing. First of all, that I want to show you to answer one of the questions is, what is it? What species of this bacteria that causing the diseases? When I'm talking about the species identification, you probably already know that most of the genetic materials that people use for species identification, it is stickiness. And stickiness is still good for using for species identification. But since, let's imagine you sequence the whole genome of the bacteria, why don't you use just stickiness? And stickiness only less than 1% of the whole information that you have. Why don't use them all to identify species? Of course, you can use them all, but it's not really computational possible to compare the entire genomes of bacteria with like 5 million base pair just to identify species. Of course, it's possible, but it can take a lot of computational cost and time. But to make it easier, what you can do, instead of compare the whole circular 5 million base pair, you chop them. When I say chop them, you cut them into small pieces. That small pieces we call Kmers. So when I say Kmers, I mean a bunch of sequences with a number of K base pair. When I say 7-mers, I mean a bunch of sequences with 7 base pair. So you have something like this, and in principle, if any genome that are similar, you chop them with the same Kmers. When you compare Kmers, they must share a lot of Kmers, and that's exactly the principle behind the method that we use for speciation. So what we can do, we collected all available known species of bacteria and chop them into Kmers and put them into the database of Kmers. Once you have your unknown sequences, you chop them into Kmers and you compare the Kmers of your unknown genome into the known Kmers in the database, and we give you predicted species. Here is example. The tool that we're talking about is called Chemifida. It's a web-based tool. You just you just upload your genomes over here, and then it's gonna show you the species. It's an example. The species of your unknown sample is actually Salmonella. Here is one very example of using a very easy tool to identify species of bacteria. The next example, how can you fight it? Before you know how to fight it, you have to know if this bacteria resistant to any of the drugs or antibiotics that you already have available. So this is maybe easier to understand, because to know if bacteria is resistant, one of the mechanisms, actually 2 mechanisms in the bacteria to be resistant, that is contained antimicrobial resistance. Another mechanism is has some mutations in some genes that make them resistant. So we actually collected genes, AMR genes, antimicrobial resistant genes, and known mutations in some genes that make them resistant. We collect those genes, put in the database that we call ResFinder database. And then we use BLAST. BLAST is just a tool to make the sequence alignment. And then it gives you if any of the sequences in your genomes contain any AMR genes. And the database that we have here is not only just randomly collected gene, we also have human curated before we actually add them in. And we also tested this ResFinder database with the bacterial strains that we actually did phenotypic tests in the lab, and then do whole genome sequencing and compare the result from whole genome sequencing using ResFinder and phenotypic testing, and we found 99.74 concordance between those methods. And it's actually very well concordance. So here an example, you can see that the 2 mechanisms that I talk about, we call point mutation mechanism and the acquired antimicrobial resistant genes. You can choose if you want to identify AMR genes from those mechanisms, and then you upload your genomes over here, and then it gives you this example of point mutation that causing resistance. For example, here it's in the gyrase A gene, and from this position of the amino acid S change to L, and then it's give resistance to quinolone. And here is the example of the having actual resistant genes. For example, BLA-CTX-M15 that causing beta-lactam resistance. Here is just an example of the result. I want to another example of using ResFinder in something that quickly responds to some of the important issue in the antimicrobial resistance. So this has happened in about almost the end of 2015. There is, I think the scientists in China that they reported a gene called MCR1. MCR1 is the antimicrobial resistant genes that make a bacteria resistant to colistin. And by the way, colistin is the last resource of antibiotics for gram-negative. What I say last resource, it means if a bacteria that's resistant to all the antibiotics that we have, normally we can kill them with colistin. And now we found a bacteria that also can resistant to the last antibiotic that work that we have, and that the gene that responsible for that is called MCR1. So since we knew that there is a gene that make this thing, make the bacteria resistant to the colistin, what we did, we included that gene MCR1 to our red spider database, and then we do screening for all the bacteria that we have sequenced in our databases, which is about 3,000 of the E. coli and Salmonella. And then we found about 5 E. coli out of those 3,000 that contain MCR1. So, and about a week, we submit this paper and it got published. And my point is, all the process I'm talking about, getting the MCR1 in our red spider database, screen 3,000 genomes, and know that we have only 5 genomes of E. coli that contain MCR1. That took about just 1 day. So this is, and imagine if you had to do it phenotypically in the lab to screen 3,000 strands, how long would it take? So this is just to emphasize about how quickly using whole genome sequencing to actually screen all of your collection of strands to see if it contains anything that dangerous in a very quick time. So this is just one example. Okay, let me go through the other example. This is, okay, now the next question. Is it something completely new, or is something that related to this outbreak or not? So all of these questions, you have to do something that we normally call phylogeny. Phylogeny is like, it's like you make a dendrogram. It's like a family tree of bacteria. You make a dendrogram to see how are they closely or non-closely related. So I just tell you one example. This is a listeria outbreak in 2014 in Denmark. And from the whole genome sequencing, they knew that if the cause of this outbreak is from ready-to-eat meat. How did they know that? How did Danish authority know? This is the Danish authority has been done this thing for quite some years already. So in Denmark, we have the public health that dealing with the human isolates, and we have the food agency that dealing with the food and animal isolates. These authorities, they routinely get the strands, and they also sequence the strands, and they share the data. Once they have an outbreak from the human strands, they compare the human strand whole genome data with all the animal and food. And then if they see any link, then they would know immediately what should be the cause of this outbreak that happened to the human. And that's what they found, what is the cause of this. And how do they know that? Of course, they need to use some kind of phylogenetic tree program. This is one example of the phylogenetic tree that building the tree by looking at the SNPs. SNPs is single nucleotide polymorphism. It's basically a point mutation in the chromosome of the bacteria. This identifies SNP and make a phylogeny out of that data. So basically, you put all of your strands that you want to do phylogenetic tree, of course, with the reference genome, and it's going to give you the tree. Okay, and just tell you another example that very much related to COVID. And I think this is a great idea of using phylogeny in the real-time tracking of any pathogens. The next strand plays a very important role during the COVID-19 tracking, because all the people in the world have sequenced COVID-19 genome and they submit to public databases. And NextStrand actually take those data and make phylogeny. And what you can see in the phylogeny, you can see the phylogeny of any specific country. You can see phylogeny from a global phylogeny of COVID-19 genome. You can compare some different countries, different regions. So this is example of the great tool to combine sequencing technology, sequencing data, put it in the phylogenetic tree in the real time, in the web-based version that everyone can access without any bioinformatics prior knowledge. Okay, and I would like to, this is if you want to hear about what in detail of all the tools and topic that I've been talking, you can go through my Coursera course. This is basically is more detail of the thing that I have talked. Okay, moving to the last topic that I want to talk today. This is just another example of beyond just using the sequencing data just for a specific study or specific species that you study, but this is used for the global surveillance of something. In this case, a global surveillance of AMR. So basically, do you want to know how much AMR that we have in the world? So of course, it couldn't be possible to get the sample from everyone, but everyone already give the sample every day, and all the sample already collected in sewage. So we basically go out through and get the sample of sewage sample from around the world in 2016, and we also continue doing it from for until now. So, but the result I show you is from 2016, and when we collect the sewage data, it's not going to be whole genome sequencing anymore. It's going to be something that we call metagenomic sequence. That means we're not going to purify any specific bacteria, but we're going to sequence everything in the sewage water. So if everything going to get sequenced and everything, that means bacteria that contain antimicrobial resistant genes. So once we sequence the, oops, sorry. Oh, okay. Since we sequence metagenomic sequence of those metagenomic data of sewage, we can use program to quantify abundance of AMR. And here's an example of abundance of AMR in different AMR classes. You can see that there are some patterns that are similar between European countries, North America, and similarly between African and Asia, and also the South America. And you can see when we do the what we call PCOA, just to cluster abundance and different type of AMR, and you can see some cluster over here. And more than that you can do. So combining the abundance of AMR from the metagenomic sequences, you can combine that with some kind of human development index data from different countries, and using machine learning, you can use machine learning to combine these things and predict how much abundance of AMR from different countries and visualize them in the map like this. So the more intense of the blue, the higher abundance. The lower intense of the blue color is a lower abundance of AMR. Okay, and I think I also run out of my time, and that's it of my talk. So now it's time for everyone if you have any questions. I'm gonna see from the chat if there is any question. No.
Thank you so much, Dr. Shini. Let's see if we have some questions. First of all, thank you so much for your presentation. It's very didactic and very pedagogic, and I really appreciated, as well, all the examples that you brought to our attention and you shared with us today. Let me see if we have yes, we have a question from Bolivia. Dr. Annette Vasquez, please go ahead.
Good morning.
Yes. Good morning.
Good morning. I want to know, well, my name is Annette Vasquez. I am from Bolivia and I'm professor and researching of the Mayor de San Andres University. We are starting working with some projects of genomics. We started working with SARS-CoV-2, but we would like to work in some networks. How can we communicate with you? Or can we do some work together?
Oh, I think you just asked for the corroboration, I guess, right? Okay, so I'm basically working with bacteria. So if you work with bacteria, of course, you can contact me. But for COVID-19, I think there is a group, if not, I think it's also another project that my colleagues is working on. It's called VEO, V-E-O, V-O. So it's a EU project. So one of the work packages in that VEO project is COVID-19. And they use a lot of sequencing, a lot of sequence analysis in their package, in their work package. So that could be the good place that you can start contacting them. And also the next trend, if you go to the next trend website, you're gonna see many people that involved in the COVID-19 genome. Mm-hmm.
Thank you so much, Dr. Shini. Maybe I give the floor to Dr. Jairo Mendez. As I said, we are co-organizing this series of meetings, webinars, and he as well leading what is the regional network for genomic sequencing of COVID-19. So over to you, Jairo.
Thank you, Andrea. Muchas gracias, Shini, por tu presentación.
Thank you very much for your presentation, Shini. And responding to the question from Bolivia, as Dr. Vicari already mentioned, we'll be talking about this later on. We, and in particular with SARS-CoV-2, which was the intervention, I'd like to remind you that we already have a genomic surveillance network in the Americas operating precisely for that reason, to facilitate collaboration between countries, but especially considering the need for this information to go beyond a simple research interest. It needs to actually find responses to surveillance in total coordination with epidemiological surveillance of events. So it's also very important to bear in mind that the best outcomes and precisely what we expect from these webinars is to have sufficient information so that countries can coordinate and integrate surveillance, in this case, for a pathology such as SARS-CoV-2, and the tools available for sequencing or data. So our invitation is that you use the official channels in your countries, and that in this case, it would be through the public health laboratories, such as the one in Bolivia, both at La Paz and Santa Cruz. So through those laboratories, initiate contact in order to achieve any collaboration, whether it be at the country level or regional level. Some people in the chat have been unable to hear, apparently. They're mentioning in the chat that they haven't been able to hear it. Let us see how to best solve that. There's a technical problem. Thank you, Jairo, for that clarification. Do you have any other questions for Dr. Shini in regards to her presentation? In the chat, we're seeing that many people can actually hear without any problems, so it might be one person with having some trouble. So if there are no additional questions or comments in regards to Dr. Shini's presentation, let me once again thank you sincerely for your presentation. And we're on schedule now to move to the next speaker, and I'll switch back to English.
So I'm switching to English to introduce our next presenter. Our next presenter is Dr. Nima Moshiri. He's an assistant professor in the Computer Science and Engineering Department at the University of California, San Diego. Dr. Moshiri works on computational biology with a research focus on viral phylogenetics and epidemiology. Today, Dr. Moshiri is going to talk to us about the application of bioinformatics in the real-time molecular surveillance of viral pathogens. And so I hope, Dr. Nima, I can give you the floor. Thank you so much for being with us today. Over to you.
Thank you. Yeah, thank you for the excellent introduction. Can everyone see my screen, I hope? My slides?
Absolutely.
Perfect. Thank you. Yeah, so thanks everyone. Thanks for being with me here today. So I'll be giving an overview of some of the applications of bioinformatics in viral pathogen surveillance. And specifically, these are things that are being used commonly in the SARS-CoV-2 surveillance. So here's a general outline of my talk. I'm going to first just introduce viral molecular epidemiology. I'm going to talk about what bioinformatics approaches occur to sequence the first viral genome of a given epidemic or pandemic. I'll talk about viral genome annotation prediction. So these two bullets are kind of what was happening at the very beginning of the pandemic. I'm going to talk about how do people sequence viral genomes in the middle of a pandemic. And then I'll have a couple of other topics related to how do you now make sense of the different viral genome sequences that you've accumulated. So first, just a brief introduction to viral molecular epidemiology. So here's an image of a phylogeny for people that might not be comfortable with the term phylogeny. It's just a fancy word for evolutionary tree. I imagine most people here have seen some form of a phylogeny in the past, but basically it's a tree structure that shows the relationships, the evolutionary relationships between species. So at the leaves, at the bottom, we have present day species. And then the internal nodes that are kind of up and above, those are the ancestors of the present day species, which we typically assume to be extinct. And the lengths of the branches along the tree denote some form of evolutionary distance or evolutionary time. So this could be human time, so it could be years, seconds, generations, but it could also be things like number of mutations, things like that. So more distance in the evolutionary tree implies more evolutionary time. So you've probably been comfortable seeing these in the context of species, but it turns out that we can also think of phylogenies in the context of viruses. So each of the leaves, instead of being different species, could be different samples of HIV, of different samples of SARS-CoV-2, whatever virus you're looking at, each leaf could be one sample of that virus. Okay, so that's some basic background on phylogenetics. Now I'm going to do a little bit of background of how do you think of mathematical models of epidemics. So thinking of an epidemic, it's the spread of some pathogen through a community, and we want to fight that spread. We can model this computationally as basically we can say that we have a contact network, which is every single possible social interaction across which the virus can transmit. So in this network, the nodes, the individuals are people in our community, and the edges, the connections between any given pair of individuals, represent some risky behavior across which the pathogen can transmit. So with HIV, these might be sexual contacts. For SARS-CoV-2, these might be just being unmasked in the same room. The structure of this network varies from pathogen to pathogen, depending on how it transmits. Okay, and we can think of a transmission network as a subnetwork of the contact network. So in the contact network, we generally had those links being undirected. So a virus can transmit either way. A transmission network is now a subnetwork of that, where again, the nodes represent individuals in our community, but now each edge represents an actual transmission event that did happen. And you'll notice that each of these branches is labeled with some time. I'm labeling them T1, T2, T3. These are the times of each transmission. In the case of a rapidly growing epidemic in which people are not reinfected yet, so if you're thinking of just short term, we assume that this is a tree structure. We assume that there's no kind of cycles in the structure. Okay, so now let's zoom in on one of these transmission events. So in this transmission event, and this is how I'm going to tie in the evolutionary history of the viruses. So if we zoom in on this single transmission event, before the transmission, the source individual had an entire collection of viral lineages within this individual's body. There was an initial source, and then that virus kind of continued to evolve over time within this person. And then at the time of transmission, we assume a bottleneck event. One lineage randomly goes from the source to the recipient and seeds an infection within the recipient. And then over time, both viral histories continue to evolve in the source and in the recipient. So now let's imagine that through public health efforts, we get to actually see both of these people come into a clinic and get sampled. Maybe we do a nasal swab if this is COVID-19, or a blood sample if this is HIV. Somehow we sample the virus. What we're basically doing when we're sampling, either by sequencing or whatever we're doing, is we're collecting a couple of random lineages in both individuals. So effectively what we're doing is constraining the evolutionary tree. There's some complete full evolutionary tree that includes all of these other lineages that we just didn't get to observe because our nasal swab didn't catch them or whatever. And basically we prune this tree to only include the lineages that we did sample within each person. And that is the true observed evolutionary tree. And then there's some ancestral genome sequence way back in time, and time moves downward in this evolutionary tree. So the actual sequences that we obtain are some function of the evolutionary process of that ancestral genome mutating over time, where mutations are inherited down the tree. So going to mathematical modeling, we could think of this as a generative process. We can think of each step as a random process that's dependent upon the previous steps. So we can come up with mathematical models to model the contact network. And then we can come up with mathematical models that are basically conditioned upon the contact network to describe the transmission network. So kind of like a probability distribution over how the epidemic will spread. Similarly, we can come up with mathematical models to describe the tree evolution conditioned upon a specific transmission network. And lastly, we can think of the viral sequences as some mathematical model that's conditioned upon the specific evolutionary tree that you have. So if we come up with these mathematical models, how is that useful to us? Well, those prior steps are not actually visible to us. Those are hidden from us. We can't see them. The only thing that we can reasonably accurately observe are the sequences. So kind of the problem that bioinformatics is imposing is, can we use those mathematical models to work backwards from the sequences? Can we somehow infer properties about those earlier pieces of information based solely on the sequences and anything else that we can infer from them? So in viral molecular epidemiology, we can use properties of the evolution of viruses to study a viral epidemic. So some questions that we might ask are, how did the virus spread across the communities or across the world? So here's a screenshot from NextStrain that kind of shows the spread of the virus across the world. What transmission clusters exist within our population? So here's a figure from a paper that shows transmission networks from local outbreaks in SARS-CoV-2. How is the virus mutating across the epidemic? We can come up, I think this is from outbreak.info. We can track in a given community, what is the proportion of the different lineages of COVID-19 or whatever virus I'm looking at over time? What is the molecular mechanism by which the virus invades our cells? We can do some form of like protein structure modeling to see what interactions we expect to occur. And are any of the mutations impacting the infectiousness of the virus? We can do simulations to see where do we predict that the mutations are going to land on the protein structure, and how do we expect those to impact the mechanism that we've inferred? And my interest as a bioinformatician is, how can I translate all of those questions into formal computational problems? Yeah, so that's kind of just some background. For the rest of the talk, I'm going to now translate some key problems that we have in public health into these computational problems, and I'll talk about some tools that solve those problems. So yeah, so now sequencing the first viral genome. This is kind of going back to late 2019. How did they sequence the first SARS-CoV-2 genome? I'll start with an analogy. Let's imagine that I have a bunch of stacks of the New York Times newspaper. It's a bunch of copies of the exact same newspaper. And imagine that I put a bunch of dynamite under my stack of identical newspapers. And now imagine that I light the fuse. It's hypothetical, so please don't try this at home. I'm an expert, so I can do it. Yeah, I light the fuse and then boom, the dynamite explodes. And what's left is all of these little tiny charred fragments of newspaper. The computational problem that I'm posing in the newspaper problem is, can I somehow use those little fragments of exploded newspaper to reconstruct what the newspaper said? So if you think about it, you probably are going to try to look for overlaps between the newspapers. We had multiple copies of the same edition of the newspaper, so we would look for different small fragments that overlap in some way and try to piece together those overlaps to reconstruct the genome, to reconstruct the newspaper. So this analogy applies to genome sequencing, because when we're doing genome sequencing with next generation sequencing technologies, we're effectively doing the same thing. We have multiple copies of the exact same genome, in this case, maybe the SARS-CoV-2 genome, and we're basically breaking it up into a bunch of tiny random fragments, and we sequence a subset of those fragments called reads. And the computational problem is, given all of these tiny fragments from multiple copies of the same genome, how do I reconstruct the original genome sequence? So this problem is known as the genome assembly problem. Fortunately for us, we don't have to solve this from scratch. There's amazing tools that exist, such as SPADES, which is one of the popular tools for viral and bacterial genome assembly. And you would basically take your raw sequence data and just feed it to SPADES and hopefully get a decent genome assembly from that. So that would be how you go from raw data to genome assembly. You would run it through an assembler. Okay, so now we've assembled our viral genome. And the question is, how do we annotate it? So what does that actually mean? So when I have the assembled genome, this is what it looks like. And I'm assuming all of the audience is thinking like, I don't know what that means. That's just a bunch of letters on the screen. What are you saying? So when we do the genome assembly, we're able to reconstruct the raw ACGT letters, or ACGU if you're dealing with RNA viruses. But it's still just a bunch of nucleotides. I have no idea what it means. So what we want to do is run some type of functional prediction algorithm. So one common tool that's used is called PROCA, where we basically take models that we've already constructed of other either viral or bacterial or whatever genomes, and we try to predict functionality, or we try to predict genes or whatever of this newly sequenced genome based on what we know in the past. So what PROCA is able to do, it's able to try to use features about genes that it knows from other organisms that are well studied to try to predict features of genes. Maybe you can predict the transcription start site, the transcription end. You can try to like predict, if I do translation codon by codon, what protein sequences would I get? Do those look like things that I've seen before? All that kind of stuff. So it has a bunch of different tools that have been trained based on existing data that we have. And you basically feed PROCA your newly sequenced genome, and it tries to predict all of these annotation features for you. So again, fortunately, we don't have to do this from scratch. We can use existing tools that do this. And then given the annotation that PROCA predicts, expert virologists can then go in and try to see, can we maybe like fine tune it? Can we revise that predicted annotation to hopefully come up with a more accurate annotation? So all of this is kind of towards the beginning of the pandemic. This is maybe December 2019, early 2020. But now let's move forward to the middle of the pandemic. So sequencing the first viral genome is incredibly difficult because you're going in completely blind. You don't know what you're looking for. But now let's imagine that it's been a few months and we're a little bit more familiar with what we're looking for. How do we sequence viral genomes of a local outbreak in the midst of a pandemic? I'm going to introduce the concept of a reference genome, and then we're going to talk about how we can leverage the reference genome to help us with this mid-pandemic sequencing effort. A reference genome is a high-confidence assembled genome sequence that is constructed as a representative example of an individual organism. And for context, the human genome was sequenced was assembled in 2001. That's when we got our first draft of the human reference genome. And since then, more and more organisms, we've gotten high confidence assembled reference genomes that we can use to help us do analyses. And I'll talk about how they help us in a little bit. So for the COVID-19 pandemic, kind of a record in my knowledge of the history of viruses or any type of pathogen was that just, in February 2020, just a couple of months after the first kind of hints that something weird was going on came out, like the first hints that like an epidemic was spreading, we were actually able to get a high confidence SARS-CoV-2 reference genome less than 1 month after the World Health Organization announced the mysterious coronavirus related pneumonia in Wuhan. So very early on in the pandemic, we had a high confidence reference genome that labs across the world were able to use to aid them in the study of this virus. So now, how does this actually help us? Well, genome assembly, it's actually a very computationally difficult problem if you go in completely blind, because you basically have to sequence extremely in depth to have enough overlapping fragments to be able to reconstruct the genome sequence. But if you already have a representative reference genome available, you could take a slightly different approach that requires much less sequencing depth and is thus cheaper to do, and in general, easier to do for higher accuracy. And specifically what we can do is what's called map reads to the reference genome. So in de novo genome assembly, so de novo means, from scratch, from new. In de novo genome assembly, such as the one that I described earlier, we inferred relationships between the reads from their overlaps. So let's imagine that I had a bunch of reads. I'm comparing the reads against each other. So I compare these two reads, I compare these two reads, I compare these two reads, so on and so forth. I'm basically comparing pairs of reads against each other and trying to look for overlaps. But if we have a reference genome, instead of comparing reads against each other, we can compare each read independently against the reference genome. And we can see where in the reference genome did this read most likely come from. We don't expect any of our reads to necessarily perfectly match the reference genome because the reference genome was from one specific sample, or maybe it's from a mixture of samples. So any mutations that exist in the sample that I'm looking at, I would have slight deviations from the reference genome, but it should be almost identical. It should be extremely, extremely similar to the reference. So again, fortunately for us, we don't have to do this from scratch. There exist many what are called read mappers that do precisely this. You give them a bunch of reads from some sequencing experiment, you give them the reference genome, and then they try to see where does each read seem to fit in the reference genome. Here's a list of some popular tools that do this read mapping. During the COVID-19 pandemic, I've been exclusively using Minimap2 for the combination of both speed and accuracy. If anyone has any questions about that, I'm happy to chat about it offline as well. But this is my recommendation. Okay, so let's say that I have all of my reads, I have my reference genome, and I've run my read mapper, so I know exactly where every single read best matches the reference genome. How can I reconstruct a genome sequence from my virus, from my sample? Well, basically, what I can do now is just kind of scan the reference genome position by position and see for each position, all of the reads that happen to overlap on that position, what letter did they have there? So for example, let's imagine that only one read happens to map to this position and it has an A. I would say that, well, the most likely letter here is an A, versus I'm going forward, maybe all of the reads here that overlap with this position of the reference genome, maybe all of the reads were a C. I would say, oh, my consensus sequence, my viral genome sequence from this sample has a C in the second position. And I can kind of just keep going position by position by position over and over again, each time just saying which letter is the most frequent at that position. and output that as the corresponding letter in my viral sample's genome sequence. So this is called generating a consensus sequence. Again, fortunately for us, we don't have to do this from scratch. There's an excellent tool out of the Anderson lab, Christian Anderson's lab, called IVAR that handles coming up with a consensus sequence for you. I've kind of oversimplified the general algorithm, but hopefully that gives a rough idea of how you would approach the problem. So you take your reads, you pre-process them, do inequality filtering, and then just map them to the reference genome, run the consensus sequence caller, and out comes your viral genome sequence. Okay, so now let's imagine, kind of fast forwarding a little bit further. Let's imagine that in my community, I've now employed this consensus sequence pipeline. So I've generated consensus genome sequences from a bunch of viral samples in my population. Now my question is, what can I actually do with this? How does this help me with molecular epidemiology? How do I actually do viral surveillance using this information? The first thing that we have to do is what's called multiple sequence alignment. So basically what we want to do is different samples that we have, maybe the beginning part was cut off, maybe the ending part was cut off, maybe there's insertions or deletions that have occurred. We want to basically line up all of our genome sequences such that we can find positional homology. That just is a fancy way of saying we want to line up all of the letters that correspond to the same exact letter in the ancestor. And then with this positional homology, we'll be able to do some cool downstream things in a little bit. So just lining up my sequences, this is a problem known as multiple sequence alignment. And basically, I want to align n genome sequences. So n is the number of genome sequences. And each genome sequence is length k. So with COVID-19, the SARS-CoV-2 genome is roughly 29,000 letters long. And n, the number of genome sequences globally, is over 3 million. So it turns out that finding an alignment that's guaranteed to be optimal has a time complexity that is big O of k to the n. And if you're not familiar with big O notation from computer science, don't worry. This is basically just saying that it's roughly some function of k to the n operations, computer operations that have to be performed to do this alignment. So even if we assume that every operation takes 1 nanosecond, that's longer than the existence of the entire universe. So that's clearly too slow to enable me to do real-time surveillance. So instead, what people have come up with are what are called heuristics. So a heuristic, it's something that's hopefully going to give you an approximate solution. So these heuristics give us approximate multiple sequence alignments, but they're much faster. So they're not guaranteed to be optimal. They're not guaranteed to be the very best alignment that you can get, but they generally have pretty good accuracy. And many multiple sequence alignment tools exist. These are 3 popular ones. And more recently, I've also come up with my own multiple sequence alignment tool called Viral MSA that is used for viruses. And I won't get into the nitty-gritty details of how it works. I'm happy to point people to the paper later if you're interested in reading. But basically, I'm able to do multiple sequence alignment of millions of COVID-19 full genomes fairly quickly, and it's orders of magnitude faster than existing methods. So if you're curious about learning about this tool, feel free to email me or ask afterwards, and I can send you a link to the paper. Okay, so now let's say that we've gotten all of our viral genome sequences, we've done multiple sequence alignment, so now we have that sequence homology that we've been looking for. What can I now do with my multiple sequence alignment? How does it actually help me with surveillance? So the last part of the talk, I'm going to talk about phylogenetic inference and transmission clustering. This is really kind of the important stuff for public health. This is where we can actually do something that gains actionable information. So really quickly, remember that the evolutionary history of the virus, there was some true evolutionary history. We have some true evolutionary tree, there's some ancestral sequence, and that ancestral sequence mutated along the branches of the tree as we go forward in time. And again, remember that none of this is visible to us. All of this evolutionary history is hidden. We can't see it. All we can directly observe are the sequences. And in this case, I've kept these little gap characters because our multiple sequence alignment gives us these gap characters. So we have a multiple sequence alignment of present day viral sequences. And the question that we have is, can we try to reconstruct the evolutionary history purely from the multiple sequence alignment of those viruses? This is the computational problem known as phylogenetic inference. So the most popular tool for doing this at the moment is a tool called IQTree2. In general, based on my own experiences, and this is by no means a thorough, rigorous computational statement, this is just based on my own experiences using it. I've typically found it to have the best trade-off between tree accuracy and tool runtime. So you generally get the best trees in the quickest amount of time for the most part. Here's the website to the official IQTree homepage, and here's the code on GitHub. And I'll share these. I think these slides are already made available, but I can send a link in the chat later if people are curious. Generally, the fastest tool, but has a bit lower accuracy than the competitors, is a tool called Fasttree2. And here's the link to the Fasttree2 website. And in my experiences, the slowest tool, but that generally has the highest tree accuracy, is a tool called RaxML-NG. And here's a link to the website. Here's a link to the GitHub repo. In San Diego, we've been pretty much entirely using IQTree2. But sometimes when we need a quick tree just to see, get a rough idea of what's happening, we'll sometimes use Fasttree2. Okay, so you take your multiple sequence alignment, you give it to one of these tools, and out comes a tree. But unfortunately, that tree is unrooted. So the models of nucleotide substitution that these tools assume, the models that they use to infer your evolutionary tree, these models are what are called time reversible. So the models cannot distinguish between forward in time and reverse in time. So the tree shape that you get, it's a topology that is unrooted. You don't know where the ancestor is placed. And also the branch lengths are in unit of mutations. Whereas in practice, to make this meaningful, we want our branch lengths to be in unit of time. So the next step is generally dating the phylogeny. So when I infer a phylogeny, the branch lengths are in unit of number of mutations. So each length is like maybe 5 mutations, or 5 mutations normalized by the length of the genome sequence, something like that. So we basically have information about how many mutations occurred from parent to child. But for public health purposes, it would be a lot more useful if the branch lengths were in unit of time. So how much time has passed from parent to child, maybe how many days, how many weeks, whatever. So I can infer a mutation phylogeny using just the sequences, but then if I overlay temporal information, so if I have maybe sample collection dates, so each of these viral samples corresponds to when some person physically came into my clinic. If I just write down what day that was, I can now use that time information that I have for the leaves to try to scale my branches to transform them to unit of time. So essentially what we're doing is estimating mutation rates, so the number of mutations per time across every branch in the tree, and then we're just kind of scaling the branches accordingly. So a lot of tools exist to do this. One of the popular tools is called TreeTime, and here's a link to the GitHub repo. It's developed by the Neher Lab. And a bonus that this tool has is that it can also do what's called ancestral state reconstruction. So not only can it determine the scalings of the branches of the tree in unit of time, but it can actually also predict what were the ancestral sequences up the tree. Another popular tool that people use is called LSD2. LSD stands for least squares dating. Here's the link to the GitHub repo for this. A less popular tool, but one that's extremely fast and is gaining some interest, is a tool called LogDate. Here's the GitHub repo for that tool. I think in practice, in general, my understanding is that people will try multiple different tools and just kind of see what the different results are for the different tools. Because each of these different tools is making slightly different model assumptions or is doing a slightly different optimization problem. Another problem of interest is assigning genomes to lineages. So a lineage is simply a subtree of the overall phylogeny. So biologically, It's all of the viral sequences that inherited all mutations up to some point in the tree. A strain is a significant deviation from the ancestral virus. I couldn't really find a firm or specific definition for what exactly is considered significant here. I think this definition was from, I think it was a WHO definition, if I'm not mistaken, but I couldn't really find what exactly significant means. But generally, it seems to have some implications about significant functional or phenotypic differences. So for SARS-CoV-2, people use a tool called Pangolin to assign genomes to lineages. And here's a link to the Pangolin, the Pangolin GitHub repository. They have a bunch of criteria for what defines a lineage. They have a bunch of criteria for how someone can submit a lineage, stuff like that. But it's basically a tool that you give it genome sequences of SARS-CoV-2, and the tool will assign each sequence to one SARS-CoV-2 lineage. And then you might also want to study the prevalence of mutations. So how prevalent, how frequent is a given mutation in my population? NextStrain is a popular tool for tracking viral mutations. I believe the previous speaker also mentioned NextStrain. It's an awesome tool. It lets you visualize the phylogeny, plus the mutations, plus demographic data from the samples. Here's a link to NextStrain, and it lets you get visualizations like this. This is a screenshot of a recent NextStrain build. And you can kind of like play around with the tree. You can look at the diversity across the genome. You can see the proportions of whatever metadata you're looking at over the population. I think I'm almost out of time, so I'm just kind of rush through. I'm towards the end anyways. Covariance is another popular tool for tracking SARS-CoV-2 variants and mutations over time. Here's a link, and some of the visualizations you can get are basically, if you have a community of interest, you can actually populate this list with which lineages you're interested in, and it'll show the proportion of each lineage over time. So you can see if one is kind of overtaking the others. It's a cool tool to play around with. Another useful tool is outbreak.info, which includes information that tracks cases, deaths, lineages, research papers, a bunch of information related to COVID-19. So you can go to outbreak.info for that information. And they have these really nice reports that tell you very useful summary information. So the last part, I'm just going to kind of quickly go through because I'm running out of time. Another problem that we can also do is called transmission clustering. So a molecular cluster is a group of persons that are linked by molecular, in other words, sequence data. And a molecular cluster is a subset of the transmission cluster. So the transmission cluster is all of the people that are in a local source of transmissions. The molecular cluster is people that we have sequence data from who are also close together with respect to that sequence data. So why is it a subset? Not every infected person gets their molecular data collected. And a molecular cluster only consists of persons in a transmission cluster who got sequenced. So when we're doing transmission clustering, we have to be conscious of people that are missing from our data set. So for example, people that are diagnosed but not sequenced. So people who are in the transmission cluster but not in the molecular cluster, or people who are not even diagnosed. So people who are in our risk network, but not even in our transmission cluster. So how do people do molecular clustering from sequences? Imagine I collect viral sequences from person X and person Y. If these two people were infected by the exact same source, the pairwise distance, which I'm marking as d of xy, will be very small. If X and Y were not infected by the same exact person, but they are in the same general group of infection, d of xy, the pairwise distance between the sequences, will be pretty small. And if X and Y were infected from completely unrelated sources, the pairwise distance between their viral sequences will be very large. So the general idea is I can molecularly link individuals X and Y if the pairwise distance between their sequences is small. So basically the general algorithm is for all pairs of individuals, let me compute their pairwise distance and put a link between them if that distance is small. And then each resulting chain of links defines a molecular cluster. So for example, here, imagine that these are my 4 individuals and these edges, these kind of putative links, potential links, are labeled with their pairwise distances. And let's say that I have a pairwise distance threshold of 0.01. So I arbitrarily decide that for my virus, 0.01 is the maximum pairwise distance to link people. I would link these pairs and my molecular clusters would be this cluster containing these 3 individuals and this cluster containing only this person. What threshold do I pick? Is that threshold appropriate for all communities? All those questions, those are excellent questions that kind of take quite a bit of research and experimentation to do. But again, fortunately for us, we don't have to come up with molecular clustering tools ourselves. There's tools such as HIV Trace, created from the Pond Lab over at Temple University, that is popularly used with HIV, but you could also apply it to other rapidly evolving pathogens. So here's a link to their GitHub repo. And you can also do molecular clustering from a phylogeny. So let's say that I have an evolutionary tree, which, as I mentioned earlier, is heavily constrained by the transmission network. If X and Y are infected by the same person, they're going to be close together in the tree. If they're not infected from the same person, but they're in the same group, they'll be pretty close in the tree. And if they're infected from completely unrelated sources, they'll be very far apart in the tree. So similarly, I can define clusters using relationships within the evolutionary tree. There's a lot of ways to define what is close in the tree. So here's just a couple of potential definitions. But yeah, so a tool that I've built in the past called Tree Cluster does precisely this. You give it a tree, you give it some criteria. So what are the objective functions that you want to optimize your clustering based on? And it'll cluster your sequences for you. So I'll go ahead and stop there. Hopefully that was a good overall summary of how bioinformatics is used in molecular clustering. And I'll yield my remaining 10 minutes for questions.
Thank you so much, Dr. Moshiri, for your presentation. Really excellent as well in showing the various potential and opportunity in it. There are a couple of questions in the chat, and I will read them so they can be translated as well. And I guess some maybe you already answered in your presentation. The first one is which mapper work best for COVID-19? And the second one is, does exist a specific database with reference to genomes of SARS-CoV-2 variants, considering the high confidence the sequences must have, mutation, indels, and so on? Over to you.
Yeah, those are good questions. Yeah, so the first question I kind of touched on. I don't know if I could say it's the best, but I have found it to be the best in my uses. I use Minimap2. I can, let me paste a link in the chat. So Minimap2, it's been extremely sensitive and it's also been very fast. So I personally like this. Other people also use BWA, they might use Bowtie2, but I've really liked Minimap2 for my purposes. And then the second question, is there a database of high confidence reference genomes? I don't know if there's a database of reference genomes per se. So there is a database of genome sequences, which is the GISAID database. This is the global what does it stand for? Global, I don't remember what the acronym stands for, but it was originally for influenza, but it's been repurposed for SARS-CoV-2. A lot of the genome sequences on this database are very high confidence, but some of them are not so much high confidence, which is why I wouldn't necessarily say it's a reference genome database, but it's just a big collection of genomes that you can then go in, and for each genome, it has some quality metrics of like, what was the coverage? What was the were there any strange mutations, any strange insertions or deletions that aren't characteristic? And then you can kind of manually curate your own collection. I also know GenBank has a collection of some high confidence genomes as well that you could potentially use as a reference. Your main criteria are, you usually want to do something de novo. So the consensus genome sequencing pipeline I described, it's useful for in the midst of the pandemic, but so the reference genome makes the computational problem easier, but it also highly guides you towards coming up with a consensus sequence that looks like the reference genome. So if you wanted a high confidence reference genome that was unbiased, you would want to sequence de novo very in depth, so very high sequencing depth, and then do de novo genome assembly.
Thank you, great. Another question in the chat, and again, I will read it so it can be translated as well. When you use reference genomes to make the assembly, do you not lose the new information? So it's moving around.
Yeah, was that the whole question?
No, sorry.
Sorry, yeah, take your time.
Do you not lose the new information that the new virus will have? In that case, is it not better to make de novo assemblies and later compare them with the reference genome? That's it.
Yeah, that's a great question. So, so the first question, do you lose anything when you use the reference genome to come up with your assemblies? Or I guess I wouldn't even call them assemblies, I would call them consensus sequences. In general, as I mentioned, doing this reference guided approach to getting your genome sequence will definitely be biased by the reference genome that you use. In general, the tool that you use to do your read mapping needs to be sensitive enough to enable sufficient differences with respect to the reference genome. So for example, if there's an insertion that is a real biological insertion, as we have seen with spike protein in COVID-19, you don't want your read mapper to throw away any reads that have that insertion just because they don't perfectly line up with the reference genome. I wouldn't say that the current consensus sequence methods are bad though, because the read mappers are generally designed for human genetics, in which case we have much larger scale differences with respect to the reference genome. So comparing viral genome sequences to a viral reference genome is generally a much simpler computational problem than comparing human reads to the human reference genome. So the read mappers are usually sensitive enough to keep track of that information. But that doesn't mean that it is necessarily the best. It's just that I think if you had unlimited money and unlimited time, the best way to do it would just be to sequence every single sample extremely in depth. The problem though is for de novo genome assembly, you need very, very, very high sequencing depth. So it's much more expensive and much more time consuming. Yeah. So I would say yes and no.
Okay, thank you. The next question is in Spanish. I will read it first in Spanish. If maybe you know Spanish or...
A little bit, más o menos.
Or you can get the translation, or we'll try to make my own translation. ¿Se tiene ya alguna predicción?
Do you already have any prediction or mathematical estimate of how many mutations a virus can have? Specifically, how many more would SARS-CoV-2 have, and in how long and how much time will these end?
So do I have a mathematical model for how many mutations are in a viral genome, specifically SARS-CoV-2? And I didn't really get the last part.
And how many more mutation we can expect?
That's a great question. Yeah. So for that, I would highly recommend taking a look at Next Strain. I'm going to post a link in the chat and I'll share my screen as well. So Next Strain, at the bottom of it, let me pull it up. There we go, share my screen. So Next Strain at the bottom of the screen, if I scroll down a bit, has, there we go. This is the plot that I'm looking for. So this plot here, the horizontal axis is the genome positions. So position 0 all the way to position 29,000. So this is the genome. And the vertical axis is diversity. And what exact metric are they using? I think that doesn't matter for the purposes of this specific question. They're probably using Shannon entropy for this. And then this is just number of events. But this tells you how many mutations are occurring roughly at every position of the genome out of all of the sequences that they've collected. So to kind of generally answer the question, what you would do is take all of the sequences that you're interested in. Maybe you're interested in your community, maybe you're interested in the whole world. And then for each position of the genome, you would count the number of differences, like the number of different letters you see at that position. And from that, you can compute Shannon entropy, which is a metric where it's a big number if you have a lot of different letters, versus it's a small number. It's actually 0 if everything is the exact same letter. I think that would kind of help you explore. As far as can we predict how many mutations we will see in the future, I don't think we can, because if you have more lineages, so the more the virus spreads, each of these lineages is mutating independently. So I think if the virus all of a sudden gets kind of contained, you would have much fewer mutations versus if the virus continues to spread, you would have a lot more. There are current tools that people are trying to develop to try to predict what types of mutations we would expect in the future. I don't know too much about those tools, but I think they take into account fitness prediction, kind of the historical abundance of mutations that you see. I don't really know too much about how they work, though.
Thank you so much. I guess it's not only a question of molecular biology, it's as well a question of how much transmission is out there at the end, no? I guess. Thanks. There is another question down here, which is, how can we deal with convergent nucleotide mutation in SARS-CoV-2 genomes? They can affect length branches in phylogeny and thus affect divergences time estimation by ML and Bayesian methods.
Yeah, that's a great question. Yeah, so I didn't talk about that, but I'm glad someone brought that up. So in the world of evolutionary biology, there's what's called the infinite sites assumption, which is basically the assumption that every single position of your genome will at most mutate once. And as the question is very well kind of pointing out, that's a very unrealistic assumption for COVID-19 because the genome is only 29,000 letters. You have more than 29,000 people infected. So by definition, at least one site must be infected, must be mutated more than once. So I will say, with convergent evolution, you basically have multiple different lineages in the tree are getting the same mutation independently. And it does throw off phylogenetic inference. The maximum likelihood tools, I think, will try to leverage information about the other positions. So if you have this one position that has convergent evolution, but none of the other positions fit properly with the historical evolutionary history, I do think they have ways of kind of correcting the estimate of time, but it will also potentially still underestimate slightly the evolutionary time. So I think the problem there is with the mathematical models that they're using. These are time reversible models that make assumptions that every single position is independent. The only solution I can conceive, which isn't currently implemented in these tools, is to come up with mathematical models of substitution that take into account convergent evolution. So basically, if you see multiple sequences that have the same mutation, having some mathematical way of kind of separating out that even though it looks like one mutation, it might be multiple mutations. I don't do too much work in that space, so I don't know what people have come up with, but I know that people are working on that problem as well. And then you would just kind of plug in those better models into the inference tool, and then it should hopefully work reasonably kind of out of the box.
Thank you so much. I think we need to conclude the session here. Thank you so much again for your contribution. There is still a question pending in the chat. I hope we can get to that. And as well, I apologize that I oversaw a couple of questions that were in the Q&A before. If any one of the panelists will be willing to go to the Q&A and try to answer the question in the Q&A section, that will be great as well. Thank you so much. So to move forward, we are going to have a break of, I would say, 8 minutes. So we will be back by the minute 40 with Dr. Marilda Siqueira from Fiocruz. And her presentation is going to be Genomic Surveillance, a key tool for public health practice and policies. So I see you back in 7 minutes, please. Please remain connected, don't disconnect, and we will be back very soon. Thank you so much, everyone.
Okay, it's a pleasure to be here with all you in very interesting seminar. And I would like to thank SPARROW for this opportunity and also for the opportunity to see previous to me so nice and interesting talks about genomics. my talk will be more practical about this genomic surveillance, and the topics of my presentation will be as a background for this genomic surveillance. I will talk a little bit about measles and influenza that are two powerful programs worldwide, and how this program gets also the impact of this genomic surveillance in different strategies that we can use it for public health, and discuss current and future achievements, challenges, and goals. So one important question that we need to do when we are using genomic and surveillance is, what is the question? So,
apologies, Marida, the connection is...
Are you listening to me?
The connection is not so good. Maybe if you can, I would suggest that you turn off the camera, maybe it may improve a little bit.
Okay, okay, if someone can turn off the camera for me, please.
Yes, I did. So it should.
Okay. Okay, so as I was saying, one important issue when we are talking about molecular epidemiology or genomic surveillance is, what is the question? For what we are doing molecular epidemiology? So we know that many factors can influence the virus genomic in terms of their variation. So the multiple replication strategies that the different viruses use, so some viruses are RNA, others DNA, some, as influenza, has a segmented genome. So the mutation rate of these viruses are also different. And one interference in this genomic variation is in a period of time of the population interferes or can interfere in this genomic variation. So when we are working in molecular in different viruses that need to have different strategies for their control, we need to understand which is the question or what is the answer that we would like to have. We start with measles now. So measles is a huge program. And since the beginning of this program, WHO organized a work group to define, to agree in the nomenclature that would be used to classify measles according to their genomic profile. So we are using this nomenclature since 1998, so 23 years ago. So the genomic surveillance is not something new. It's something that has been used in different programs for a long period of time. And in this measles, this genomic is important to track transmission and to know if a case is imported or endemic related. So after some years of this program, after many strategies that were used to control measles worldwide, PAHO, as Americas, was a very successful program here for measles elimination. PAHO recommended studies to verify elimination. And one of the criteria, the molecular epidemiology was recommended as one of the criteria to verify measles elimination in the Americas. So when we look at the distribution of the types in different years, for example, that in the year 2010, we had many genotypes circulating in different countries in the world, different genotypes circulating. But after extensive mass vaccination campaign, we are observing a reduction in the number of the. So nowadays,
looks like we have lost the connection. Just a second, please. Marilda, unfortunately, I see that you're still connected, but we totally lost your screen and we don't hear you. Apologies, everyone. Just a little bit of patience. We try to see whether we can reconnect with Dr. Siqueira. So I see that Dr. Siqueira is still connected, but unfortunately, we cannot hear or see your presentation. So I guess if we cannot fix it in the next few moments.
Dr. Vicari, we lost her. She shows us connected, but I think we should move on to Dr. Ian until she can connect again.
Oh, okay, so we'll do that. I'm very sorry with Dr. Marilda, first of all, but for everybody as well. So I would guess we can move on to the next speaker. No, unfortunately, it looks like we cannot reconnect. So I would like to move on to the next speaker is Dr. Professor Ian Goodfellow. Is a welcome senior fellow with the and professor of virology and deputy head of the Department of Pathology at the University of Cambridge. He's a molecular virologist with over 25 years of experience working on a wide range of human pathogenic viruses. It's very nice to have you, Dr. Goodfellow, today. And his presentation is going to be about field deployment of sequencing technology, the practical consideration and solution. Over to you, Dr. Goodfellow. Thank you so much for being with us today.
Hi there. Good morning, good afternoon to everyone. I'm just going to share my screen. Let me just see if I can do this. Okay. Okay, I think that's visible. Can you see my presentation?
Absolutely, it's perfect. Go ahead, please. Thank you.
Okay, well, good afternoon, good morning. So the purpose of today's talk, I'm going to try to give a bit of an overview of a sort of personal experience of using genomics technology in the field for epidemic response. And I'm going to talk about the journey of how we went from our initial deployment of genomics in Sierra Leone, and this is just one of several large trucks of equipment that were required, and how we refined this down into a handful of suitcases that we've now used and field tested for the sequencing of pathogens, particularly viral pathogens of humans. I should start by saying that actually the vast majority of the work that we do in my laboratory here in Cambridge is not genomic sequencing. In fact, we study viruses that cause gastroenteritis, and we study basic biology of these pathogens, trying to understand host factors that are important for norovirus replication in cell culture. However, in November 2014, I was heavily involved in the response to the Ebola epidemic in West Africa. And this was primarily as a volunteer. I volunteered to work with the UK government, and I was sent as one of the first teams into Sierra Leone to set up a diagnostic laboratory in an Ebola treatment center. This is me. And we set up this testing diagnostic center in Makeni in Sierra Leone. And it was really from that point that we ended up getting involved in genomics. Essentially in December 2014, once we'd established the diagnostic testing laboratory, through discussions with a number of people in Sierra Leone and outside Sierra Leone, in WHO and some of the other agencies that were responding to the outbreak, we identified that there was a real need for genetic sequencing of Ebola virus in this epidemic. Whilst there had been, many, many thousand cases at that time in December, there were very few sequences available. In fact, it was I think that the only report to date at that time in the epidemic was in September, where there were about nearly 100 genomes sequenced, and they were from very early on in the epidemic. There was also limited access to data for those in the field. So people on the ground really didn't have much access to the sequencing data. And actually the data that was being produced in a time frame that was of little use. Effectively, the sequences were not being released. So somewhere in the region of 3 to 6 months from the sample collection to the sequence being available for analysis. So the sorts of questions that were immediately obvious that could be addressed if we had more effective sequencing of Ebola during this particular epidemic was really initially to try to confirm epidemiological links. Now, this was particularly critical during the final stages of the epidemic when there were atypical transmission chains. So whereby Ebola virus had transmitted from a persistently infected individual to somebody else. It was very important to rule out any other sources of infection, and this could be relatively easily done via sequencing. We wanted to really understand virus evolution, as we're seeing with COVID-19. When a pathogen moves into the human population and goes through repeated human-to-human transmission events, there is a possibility that it will adapt. There's also a possibility that the sequence of the virus will change so that the diagnostic tests become less effective. And without good, effective sequencing and surveillance of the pathogens of the virus during the epidemic, there was a real concern that we might be missing something and the virus may be evolving in a direction that was not going to be beneficial for diagnostics in the long term. So naively, in around about December 2014, we figured that rather than trying to move samples out to the country, which had become pretty much impossible, we thought we would just simply move a sequencer to the treatment center. And I say this was rather naive because at the time we had no experience in doing this. I had only ever sequenced bacterial species using manual sequencing during my PhD, and in the laboratory, my laboratory had no experience in doing this. So it was somewhat naive to think it was relatively straightforward, but suffice to say, we did achieve it, and we learned a lot, and we've come a long way since our time back in 2014 and 2015. So the first question we had to ask, and this is something that is very pertinent to when you're trying to select a sequencing platform for deployment into an unconventional environment, is you need a machine that's robust and tolerant of these extreme temperatures. Most sequencing machines will work fine in an air-conditioned laboratory where you can control humidity and temperature. Now, if you put them in a very humid tent in the middle of a treatment center, this is well outside its certified operating conditions, and many machines will just not function. In this particular instance, we needed a machine that was very easy to install and capable of working out of the box, because we did not have access to engineers, and none of the suppliers that we were working with would be willing to send an engineer to work with us. We needed something that was low maintenance, that we could fix if it was broken, that the workflow was simple and robust, but we would generate high quality data, and something was reasonably high throughput, because at the time, as I said, there was very little sequence data. And as we've heard in some of the previous talks, you really do need reference sequences if you're trying to do genomic epidemiology. You need a reference database to map your sequences to. And finally, we needed a company that was willing to work with us on the project and fully engage and try to help us overcome some of these real technical challenges that you encounter when trying to do sequencing in these resource-limited environments. And in the end, through as a result of a series of conversations, we settled on a Thermo Fisher ion torrent sequencer, which is quite old technology. Even by 2014, 2015 standards, this was relatively old technology. However, it was incredibly robust, and we were confident that we could get this to work and it would not be affected by the environment in which we were placing it. We coupled this with a liquid handling robot called an ion chef that would handle some of the sequencing, but some of the final stages of the library preparation, but actually most of it was still manual. And with this in mind, myself and my postdoc set about trying to move all this equipment into a tent, which was in fact a store tent in the Ebola treatment center in Sierra Leone. Because of various delays with funding, etc., and getting the equipment, it wasn't until March 2015 that we deployed to install the sequencer. And in fact, this is just some of the equipment that was used, that was brought in for sequencing. I think we had 1.5 or 2 truckloads of equipment and reagents in order to set up this sequencing facility. And here's my postdoc and I installing the sequencer. Now you can see from these images that whilst this sequencing setup was in a tent in an Ebola treatment center, it still very much looks very familiar to a standard laboratory setup. You know, we have standard laboratory benches, we had a water purifier for making ultra pure water, we had PCR machines. So it was very effective and it worked very well. But it was somewhat expensive. I'd say it cost in the region of £450,000 all in. And over the period of where we operated in the treatment center, we processed in around 1,200 samples, producing around about 600 Ebola virus genomes, which was 1/3 of all the genomes produced from this outbreak. And at the time, this was probably one of the best, if not the best, sequenced outbreaks for any pathogen. Around about 24% of the cases from 2015 onwards was sequenced, and around nearly 5% of all cases across the entire outbreak in Sierra Leone. So this was a very fruitful and very useful exercise, and we were able to set up a real-time sequencing workflow where we work alongside WHO, CDC, and a number of other agencies to provide rapid sequencing of Ebola virus and rapid interpretation of the data. We'd have weekly teleconferences with WHO to discuss individual cases. And here are just a number of examples where the information was used, looking at cross-border transmission. We were identifying links in particularly hard to investigate cases. This was particularly where we had reemergence of viruses in different regions of the country, particularly those where there had not been a case for quite some time. We were then able to link back to survivors and confirm that this was not a case of missing an ongoing chain of transmission. In fact, this was just a virus that had persisted in the survivor and then subsequently reemerged in one of their contacts. We also were able to provide the first evidence that Ebola virus can survive more than 500 days in an infected individual and then transmit and cause disease. So what lessons did we learn? As I said, we were completely naive to this. This was our first time we ever did sequencing of viruses in the field. We'd never even used this equipment, and we only had 3 to 5 days training on the sequencer. So what we learned was that really preparation is key. Trying to think about worst case scenarios, or where, what are the things that could stop your ability to generate data. I think these cartoons are particularly relevant. Some of the most simplest things become particularly frustrating. Trying to generate distilled water to feed into a water purifier, for example, becomes rather complex when you're in the field in a resource-limited environment. When generating the data is really critical, and in this instance it really was, it was clear that over-preparation was better to ensure that you had redundancy in all processes, so you had no single point of failure. So if one piece of equipment broke, where possible, you had an alternative. In our case, we had that, with the exception of the sequencer itself, which was so expensive, we couldn't afford to buy two. And then finally, one thing that became very clear is that process controls are absolutely critical. Ensuring that the data you're generating is of high quality, and that ensuring that the data you generate is going to be of use and picked up by others in the field and analyzed. It's really important to ensure the right controls are produced and made available to others. So what did we learn from this experience? Well, I think I can hear somebody speaking. Is that right? So we learned, I'm just going to go back one slide. What did we learn from this experience? Well, we learned quite a lot. We learned that actually it was possible to deploy sequencing into the field, and that it does have utility, but that actually there's a number of bottlenecks that make it a bit of a challenge. Now, if you look at the epidemic curve for the outbreak in West Africa, you see that we didn't deploy until April 2015, March, April 2015, which was actually well past the peak of the epidemic. So we asked the question, what if we had all these protocols and processes pre-established, and we were able to deploy earlier in the epidemic? Could we use this information, and how could we best use this information? And this is which led to the formation of the Arctic Network, which is an international collaborative network set up with ourselves, individuals in a number of universities, but the lead applicants are in Birmingham and Edinburgh, whereby we essentially were trying to build off our experience and then develop an end-to-end solution that would allow us to go from sample all the way through to...
I didn't get that. Could you try again?
Excuse me, I'm trying to turn off my... somebody is messaging me from another meeting I'm supposed to be in. So to try to go from all the way from sample all the way through to sequence and analysis and interpretation in the field in the fastest way possible. So this builds on a number of core expertise. Our expertise is in the field deployment, but also the expertise of our colleagues in bioinformatics and so on. So the Arctic network was established a number of years ago now, and as we've really seen with COVID, the protocols and procedures we've established have become quite key to COVID sequencing globally. But I just want to talk you through the processes that we've established, and this is a highlight our revised sequencing workflow, which is essentially all the equipment required to sequence viruses contained within these 4 bags, along with one cold container, which needs to be shipped alongside. This is a welcome collaborative award with University of Edinburgh, Birmingham, and some others. And all the protocols, bioinformatic pipelines, reagents, the phylogenetic schemes, primers, everything is made freely available online. And in many instances, we will provide individuals with primer sets and have supported sequencing in various places. This is a picture of our setup in DRC in 2020, or end of 2019, and then early 2020, where we trained a number of individuals to use it for Ebola sequencing. So a number of key considerations. I'm just going to walk through the setup that we have now and highlight some key features of it, and that we think are particularly important when you're deploying sequencing into resource-limited environments. The first is portability. You need to be able to carry in and pack, unpack, and repack, if necessary, the entire set of equipment relatively quickly. In the case of the Ebola epidemic, it was particularly important because the region where the epidemic was taking place was a conflict zone. It needs to be have high usability, which means it needs to be simple and robust and easy for others to pick up and use. We need a process that really maintains sample integrity and provenance. So we need a process that enables us to make sure that there is no contamination through the sequencing pipeline and that samples don't get mixed. So rather than stick with our existing technology, we decided to build off the experience of others, my colleagues that were also part of the Arctic network, which is Nick Lohman and Josh Quick from the University of Birmingham. And they had deployed a nanopore sequencer, as you can see here, into Guinea during the Ebola response. Now, for those of you who are not familiar with it, the Oxford nanopore sequencing is a small USB-powered device. It's very, very inexpensive. And the way it works is essentially in this flow cell here, you have a number of individual cells in which contain a membrane. On top of that membrane, there are little pores. And essentially, as the DNA is sequenced, it's pushed or pulled through these pores, and it results in a change of current. And this signal is then converted into DNA sequence. This is a particularly good technology to use in resource-limited environments because it is very cheap, it's very robust, it's very easy to teach people how to use this. What you will see, however, is that people who favor other technologies, which are actually often not suitable for field deployment, will quote the error rate of nanopore sequencing as being its downfall. Now, it's fair to say that the error rate of nanopore-based sequencing is indeed higher than that of Illumina and other technologies. So a single read accuracy is around about 98.3%, although that's improving considerably over time, and there's new technology which improves this quite significantly. This is only single read accuracy. In reality, you sequence every part of any viral genome multiple times to generate a consensus, and the consensus is greater than 99.9% consensus. So for almost all applications, not every, but almost all applications, and nanopore sequencing is quite capable of generating a consensus sequence that is useful for genomic epidemiology. The advantages of this technology over others is that it's zero maintenance, it's low cost, it can be very fast runtime. So from loading the sample onto the machine, it can be anything from 2 hours to 12 hours, depending on the number of samples. For 96 samples, you would typically run between 6 to 12 hours. We can run 96 to 384 samples in an individual flow cell. And you can run the flow cell for as long as you want. So you can start it and stop it. We have even had to recover samples from the flow cell when things have gone wrong. New protocols even allow you to reuse flow cells. So if you find you have 24 samples one day, 24 samples the second day, and so on, you will sequence one set of 24 samples, wash the flow cell, the next day load the second set, wash the flow cell, and so on. And this enables you to keep your costs down and reduces the amount of consumables that you need to fly in, carry into the environment. We have developed a number of sequencing protocols which are now widely available. One of the challenges of sequencing pathogens in the field is ensuring you get sufficient data generation. Now, for nanopore sequencing, we built off our combined experience in using the Iron Torrent AmpliSeq workflow and a PCR-based amplicon sequencing protocol that was developed by Josh Quick and Nick Lohman for Ebola. And we combined the two and came up with this multiplex PCR protocol, which is now widely used for numerous pathogens. And we would estimate somewhere in the region of 75% of all COVID sequences generated to date have been generated using primer sets and protocols developed by our consortium. The basic concept is that you take the pathogen of interest, in this case, let's say SARS-CoV-2, and you generate primer sets that will produce small overlapping amplicons. In this case, these are in the region of 400 to 500 base pairs. You then will use a piece of software which has been developed by our colleagues in Birmingham. This is called Primal Scheme. I'll show you, this is a simple website where you upload an alignment, you set a number of parameters, and it will very quickly give you a scheme design. This will then, in the case of SARS-CoV-2, it generates two sets of 48 amplicons. Essentially, we take a sample, we split it into two, we do two PCR reactions, which are then subsequently combined and through a process of purifications and ligations, will end up with a sequencing library that's suitable for loading on the flow cell. We've significantly streamlined this protocol now, so it takes probably in the region of less than 12 hours. You can do it significantly faster with fewer samples, and we've significantly reduced the cost. But these protocols are now widely available, and as you can see here, they've been independently validated by other people who are working on genomics in the field. Maintaining a clean workspace in the field is absolutely critical. I see a lot of people publishing data where sequencing has been done in resource-limited environments without any mention of the controls that are used to ensure that the sequencing is of high quality. And when you come to analyze this data, it's often very clear when there are problems with the data. And I think this is something people really need to take into consideration. Now, a typical approach in a standard laboratory is to use a PCR cabinet, which gives you an airflow as a stainless steel interior that can be UV decontaminated. It's incredibly expensive and they're incredibly heavy, around about 30 kilos per cabinet. Now, unfortunately, there is no low-cost portable PCR-specific cabinet being designed. However, through a series of trial and error, we eventually found hydroponics grow tents, which are actually used for indoor growth of of plants, frequently used for the growth of recreational drugs, I've been told. That these can be purchased for relatively low cost. These are around about £47 here in the UK. And these provide a very robust, clean working environment for doing sequencing in resource-limited environments. And in fact, we've used these repeatedly. We have used them here in my laboratory in Cambridge for COVID sequencing for the last 18 months because we can essentially dispose of them when we're finished, and they're very easy to put up, very easy to collapse, and very inexpensive. Monitoring contamination through the sequencing process is absolutely critical to ensuring you're generating robust data. Every single batch of sequencing should include an extraction control, which is extracted at the same time that the samples are extracted, and an RT-PCR control, which will through the entire process, and sequence is generated on these at the end. Failure to do so means that you are not generating sequence which you can be 100% confident is clean. We've also tried to establish a workflow that minimizes power consumption. Whilst most environments we work in have access to mains power, this can be somewhat unreliable. So we prefer to use lithium-ion battery packs for most of the equipment where possible. And in fact, pretty much everything we do now for the sequencing can be run off lithium-ion battery packs. As long as you can recharge these at the end of each day, you can do everything without mains power. You do, of course, need access to a cold storage, but there are other solutions that work for this. This is one of the cabinets set up for sequencing. We use small magnetic USB-powered rechargeable lights. The equipment, which is just really a centrifuge and a vortex. We have higher powered centrifuges that also run off these lithium-ion battery packs, and we use a lab mat. Another key feature we use for decontamination are these small USB-powered rechargeable UV lights. We would put 2 or 3 of them in these cabinets at the end of each run, followed by a 1% sodium hypochlorite bleach solution as a decontamination step. These two things combined give you a very powerful and very effective contamination control process, which we've been able to use here in Cambridge for many months without any real problems. The amplification process, of course, requires a PCR machine. Now, you can, of course, carry large PCR machines to the field as long as you have a good power source and you can afford them and you're not too concerned about, you've got sufficient redundancy in the system. We've tended to opt for these low-cost PCR machines, primarily partly because of the cost, but also to include some redundancy. So that if one of the machines breaks, we have another or several other that we can fall back on. We found them to be very, very useful and very, very robust. They're around about £700. You can run 16 reactions, which is in fact 8 biological samples per machine. We can run them off battery packs when required. And whilst they do not have active cooling, which is required for some of the steps of the sequencing, you can quite effectively chill these down if you have access to ice in bags or freezer blocks. So we would often just freeze these elsewhere, carry them to the site on the day, and then store them in a polystyrene box and bring them out to reduce the temperature of the PCR block when required. One thing is absolutely critical to ensuring effective minimization of contamination in the field is a directional workflow. Now, many of you will be familiar with this in terms of running diagnostic laboratories. I would always recommend that you do not try to attempt sequencing in the same physical space where diagnostics is run, because the amount of DNA amplicon being generated during sequencing is very, very high. And there's a lot of physical manual manipulations which significantly increase the risk of contamination. So we would always recommend running sequencing in a completely independent physical space. You can, if possible, run each of these steps in separate rooms, but we find that often there isn't sufficient space available. We can run this in a 4 by 4 meter room, as shown here in the Research Institute in DRC, and also in situ in a facility in Goma. So we set up 3 cabinets with 3 slightly different sets of equipment. We have a sample extraction and sample addition. Our preferred mode of operation is to work with the diagnostic laboratory who will inactivate and extract the sample for us. If that is not possible, then what we can do is either inactivate the sample at source. So following sample collection, we inactivate it while it's being collected, and then we can bring it into the sample extraction cabinet where we would extract it before moving it through the process. Alternatively, we would set up a completely independent cabinet in a completely independent room, which is a containment, provides some biological containment. These don't provide any biological containment. They only provide clean working space for the molecular biology manipulations that we do as part of sequencing. So if sample inactivation and extraction is required, we have another solution that we place in a different room. Something we found particularly useful and quite transformative when trying to run sequencing in the field, and this massively overcomes any problems around compatibility of laptops and installed software, is that we have generated, or the Arctic Network have generated, our collaborators in Birmingham, a lab on an SSD. And essentially, these are pre-configured solid-state drive images that can be downloaded, installed onto a relatively cheap solid-state drive, and then subsequently plugged into a suitably powered laptop. Now, if we're doing base calling, we would need a GPU-enabled laptop. You can then boot from the solid-state drive. It has Linux. It will have every single piece of software you need to go from sequencing all the way through to some sort of basic analysis pre-installed on the solid-state drive. And this is one of our colleagues in Goma in DRC actually using our SSD for Ebola sequencing and installing it as shown. We found it's also pretty important it can be very useful rather to monitor the sequencing in real time. And our colleagues in Edinburgh have developed a piece of software called Rampart, which will work alongside the Oxford Nanopore technology to give you real-time mapping of the data. This is a free piece of software which can be easily downloaded and installed. And essentially, this is SARS-CoV-2 sequencing. As the sequence is coming off the sequencing machine, it will be mapped to a reference genome and the read counts per sample shown here. You also get, for each individual sample, you'll get a coverage plot, which shows you how effective the sequences have been. And you'll also get some sort of estimation of how long it will take for you to reach 99% coverage across the genome. We found this to be an incredibly useful piece of software for monitoring the efficiency of sequencing, because you can quite rapidly identify when there's been a problem, when there's been possible source of contamination, or when an entire library has failed for one reason or another. You can go back and restart the sequencing sooner than you would with other technologies. So what do things look like when they go wrong? So this is evidence of contamination. So again, we use this in this is a Rampart plot. So this is mapped reads across the SARS-CoV-2 genome. This is a good clean run where we have an extraction control, which is included in the batch, which has been sequenced. You will often get some amplification, but this is just noise. It's non-specific sequence pieces of DNA, which are basically amplifying. They do not map to the viral genome, and they're completely absent. And when we look at the RT PCR control, again, we get, in this case, we have 2 reads mapped, which is well below our threshold, which would be anything above 20 reads mapped anywhere in the genome, we would fail the run and repeat the whole process. And here's an example where we have a failed run, where we have evidence of contamination. You can see clear evidence of reads across the genome in our extraction control and the RT-PCR control. And in this instance, this particular batch of samples was re-extracted at source and passed. So this is what the equipment looks like, packed up. One thing, this sort of plays to my sort of rather obsessive nature in labeling things. Something we found very important when you're deploying these sorts of technologies to the field repeatedly, packing them up, moving them to places, is having an effective labeling system. It may seem silly, but having the wrong cables or not be able to find the right cables at the right time can mean that your sequencing trip is completely destroyed, or you need to waste 2 days traveling around the country to find a very specific cable to connect one piece of equipment to another. Packing things in a way that makes them very easy to identify, using packing lists is critical, so that you make sure you don't leave anything behind. So how does this workflow compare to our previous workflow? I'll remind you that previously we'd spent around £450,000, and over the period of 8 months, we'd produced 600 genomes. Now we, here in Cambridge, back in March 2020, we were asked by the UK government to start sequencing COVID. Because we had developed the Arctic workflow and the Arctic field deployment equipment, we essentially just used the same workflow, even though we installed it in our traditional lab. Unfortunately, at that time, we had no equipment or reagents because we had left them in DRC with our collaborators. But very rapidly, we were able to piece our kit together, and within 3 weeks, go from nothing to in excess of 600 genomes. This is something that took us 8 to 9 months and nearly £400,000 back in 2015. And this work has now then continued on, and we produced in excess of 15,000 sequences in Cambridge alone, and we've worked collectively as part of the large COG UK network. One thing I think that it's really important to consider is the time from sequencing to upload. This is a piece of information I found recently where somebody has taken the time to look at date from sample collection to depositing on a public database. And you can see there's a huge difference as you go through the various countries doing sequencing, with the UK coming out pretty well in this. And I think this is probably just because we have automated processes set up now to push sequences to GISAID. What I would like to say is that generating sequence is actually just the first step in the process. It's really essential that data is shared rapidly for analysis, and the sequencing data itself is almost worthless if you don't have good quality metadata linked to it. Just looking at sequence of pathogens without knowing where and when it was collected, and ideally some other information about the individual patient, it really reduces the value of that data. So in summary, just to wrap up, I would say that probably most practical issues associated with rapid viral genome sequencing have been solved. Of course, each environment in which you try to deploy this technology will have its own particular challenges. And of course, each pathogen brings with it another challenge in terms of sample diversity and so on. There are real issues around data analytics and data sharing. Some of these are practical and some of them are political. Data sharing is really important for making the most out of genomic information, but there are clear cases where scientists in the field are abusing this free access. And I think this is something that needs to be very considered. Sequencing of viruses has a real role to play in epidemic response, but its utility will very much differ from epidemic to epidemic. It is not always the case that sequencing the virus will be of any use. You need to consider carefully the questions that are trying to be addressed and the nature of the pathogen that you're studying. And quite critically, I would really hit home this last point, that effective linkage to patient data and epidemiological information is really critical for extracting the full utility of any sequencing platform. So finally, just to thank the people who are involved in the work. All the work I'm going to spoke to you today was developed by the Arctic Network and then subsequently deployed and used as part of the COG UK. And with that, I'm happy to stop and take any questions.
Thank you so much, Dr. Goodfellow, for your presentation. Really excellent to see as well the practical implication, in terms of field deployment as well. There are a couple of questions. Some of them are actually in the Q&A facility, and maybe I will start from there. There is a general question that was actually from before, but I think it's very much related to your experience as well. I will read it first in Spanish so it can be translated. ¿Cómo se aplica y se relacionaría?
How does one apply and relate the molecular study with epidemiological research having to do with the outbreak studies? At what point in time would one have to use the study?
How are molecular sequencing methods applied or related to epidemiological investigation of outbreaks? At what moment they are best used? I think you have hinted a little bit in your presentation, and maybe if you can return on this point. Thank you.
I think what's really important here is to accept that the people who are often generating the sequences, like myself, are often not the best people to decide how that data is used. And it's really important that those sorts of questions are had prior to even starting sequencing. It's quite frequently I would see that, we'll just sequence stuff because that's what everybody does. It's really important to sit down with the epidemiologists and to say, and the WHO and the people who are responding to the outbreak, what information do you need and how can we use our expertise? So I think what's critical for me and going forward is really to try and encourage people to have those open discussions prior to even starting, because if you're not collecting the right metadata, the sequences you are generating are of very little use. So I would say right at the start, ideally you have those discussions before an epidemic even starts. You already consider what each pathogen, the information you might need to be collecting for each individual pathogen.
Thank you. I will read another one, which is as well in the Q&A facility. It's in Spanish. I will try to do a translation on the fly. In the experience of a field lab and working with viruses like Ebola that are highly contagious and they need a high level of biosecurity. How did you achieve and maintain biosafety to work in the field with Ebola virus, and how did you deal with the infectious waste? Yes, thank you.
So in most cases, as I said, we try to partner with an existing diagnostic laboratory. If you want the sequencing equipment to fit into 4 cases, you need to partner with somebody who is providing that sort of upfront extraction, inactivation and extraction, ideally for you. There are solutions in the case of Ebola in Sierra Leone, we used flexible film isolators. Now, they can be deployed to the field. They are not particularly portable. There are some portable versions of them that have been used by the European mobile laboratories, but they are quite small and quite restricted. So we have a solution that works, it's a commercial solution, but our focus has primarily been always try to partner with an existing, to bolt on our sequencing approaches onto an existing diagnostic laboratory. The infectious waste, in the case of Ebola virus, was essentially incinerated in a fire pit. And most things were decontaminated with chlorine, sodium hypochlorite. But where that was not possible, things were double bagged and then incinerated through an incinerator that was available to us on site.
Okay, thank you so much. Another question in the chat is in Spanish. I will try to translate it. How long after you start sequencing is the rampart analysis done to determine the efficiency of the library?
It's instantaneous. So you basically, you load the sequencer and you press go. So I would say it starts looking for sequence immediately, but it will not map sequences until it's generated. I think it's 5,000 reads per sample, or is it 4,000? Essentially, the software, the nanopore software, will output data in batches. It will throw out sequences, I think in 4,000 reads at a time, and Rampart picks those up and maps it. But we find typically it's within 5 minutes of starting the machine, you can get Rampart and you'll see the library running. So certainly within the first 15 minutes of loading your sample, you will know whether or not things are have worked. Within the first hour, you will know whether all samples have worked or not.
Great, thank you so much. Another question as well in the chat, always in Spanish. I will try to translate. How do you manage the short duration of the flow cell, of the nanopore flow cell, 3 months, durability? How many times approximately do you wash them and do you reutilize them, the flow cells? What is the quality that you accept in the sequences of the transmission?
Yeah, the time frame, of course, it's a challenge, isn't it? You know, we try to ensure we have some flow cells with us, but even once they're past there, if they're stored in an appropriate way, they last considerably longer than the manufacturer will tell you, which is 3 to 6 months. I think they guarantee them for 6 months in the UK. But they will last longer. We've used flow cells that are over a year old, and it works perfectly well. But that is an issue when you're taking it into the field. You want as new a flow cell as possible and ensure that it's stored. The second one was how many times we rewash. Now, this depends on your question. So for SARS-CoV-2, we have 96 barcodes that we would run on a single flow cell. But we can run those on 1 go, or we can run it in 3 goes, or we can run it in 4 or 5, but we would never run more than the 96 barcodes, because even though we can wash the flow cell, we would never want to reuse that barcode, which is essentially the identifier for the sample, on more than one biological sample. So we work through the barcodes, which are what we assign to each individual sample. And once we've used all those barcodes on that flow cell, we would no longer use that flow cell. However, we have used other pathogens on the same flow cell. So we've sequenced 96 coronaviruses on one flow cell, washed it, and then sequenced herpes viruses on the same flow cell. Because you can remove, it essentially removes the DNA. We wash it with DNAs. But the reason we do not use the same biological sample with the same organism is because sometimes there's a very minor amount of DNA which remains in the flow cell you cannot get rid of. But if you're sequencing a completely different pathogen, you can reuse it. Typically, we find the flow cells won't, maybe up to 4 or 5 reuses, and then that you have fewer and fewer flow cells. Pores, sorry.
Thank you, excellent. A last question, it's in English in the chat, regarding the use of sequencing using Grion. How did you solve the genome analysis? How did you solve the internet needs?
So the internet needs you can get away with. There's a very simple line of code you can install on Gridiron that Nanopore will tell you, which will basically because the software normally looks for an internet connection continuously. But there's a piece of software, there's a line of code you can run that prevents that, and Oxford Nanopore will tell you that. For the genome analysis, we install all the software on the GridION itself. The advantage of the GridION is a very powerful machine. So we run all the assembly on the GridION. And then once the genomes are assembled, it's only 30,000 individual letters. We can email those sequences to anyone, even via a mobile phone. It's relatively straightforward. But the base calling and the assembly and some basic analysis we can do on the GridION using the compute power of the GridION just by installing software.
Okay, excellent. These were all the questions that were out there. There is actually a last one. What is the quality value that do you consider acceptable for the sequencing?
Depends what you mean by quality value. Essentially, when we're sequencing SARS-CoV-2, we would use a minimum number of reads per nucleotide. We would want to read each individual nucleotide a minimum of 20 times, ideally 50 to several hundred. So we would sequence every single part of the genome a minimum of, minimum of 20 times, but I would say typically it's several hundred times. In terms of coverage of the genome, ideally we get somewhere in the region of 99.8%, I think, of the entire genome sequence with our new primer sets. But in some instances we get, if the viral load is low, we get smaller, we get fragmented genomes. But that sequence in itself can still be useful depending on the question you're trying to address. So fragmented genomes in certain circumstances can contain information that is still useful.
Yeah, thank you so much as well for your contribution today. It was really great having you as well. Let's try to go back and see whether Dr. Sikira is able to connect again and to retake and finish her presentation. Hi, Marilda. We see the presentation, but we cannot hear you.
So hello, can you hear now? So
we can hear you, yes.
Yeah, so so first of all, I apologize for this. Well, we are always having challenge in our life, and so this is one of them. So I will continue more or less when I think that you stop hearing me. So I talk a little bit about that this genomic surveillance. We need to have a question for that, for public health issues, for what we are doing this. So I'm using 3 models of this, that is measles, influenza, and after SARS-CoV-2. And so I explained that for measles, WHO developed a nomenclature, and we use it in a very successful way in the measles elimination program worldwide. So, and based on the success of the vaccination program, mainly in PAHO region, there was some criteria that PAHO recommended to include in the document for verification of measles and rubella elimination. And in this document, molecular epidemiology is one of the 5 issues that we need to present, to consider as measles eliminated. So it's a very practical issue. In this case, the molecular epidemiology, the genomic analysis in the world. So as we can see, the measles over the times, that 24 genotypes presented different distribution in the different countries. with this, we were allowed to say, oh, this is endemic in this country, or this is a sporadic case, this is an importation. So the genomic is a very important tool to help the understanding the strategies used by the different countries. So we had a huge number of genotypes in previous years, and now nowadays, just 3 genotypes circulating in the world, with the genotype H1 restricted mainly to China. So we can see here one example how we use in the field this genotype during some years, like 2005 and 2006. So the identification of genotypes imported in different countries. So all these issues here are a strong work by the epidemiological surveillance, try to track, to understand where the case, the sporadic case or the outbreaks, where the source of those outbreaks, and this genotype information were absolutely useful for this understanding, for the comprehension of this importation or not. So I can give here, show here one of the example that happened in Brazil, that we had in 2005 a sporadic case, and when we track it, we demonstrated that this person was in the South Asia, and all the way that the flight had and which genotype we had from that case, and the measles transmission followed that imported case. We, with the classical epidemiological, with the people on the field, were able to track it and to show that this case do not give a chain of transmission, a long chain of transmission, and all the other cases from this one belongs to the same genotype. So is genotype disappearing for measles? So we started with 24 originally recognized genotypes, and now we have mainly 2 genotypes. So this reduced in diversity is giving us also some challenges in interpretation of the different, mainly sporadic cases that we are found in the different countries. So how can we use the sequence, the genomic information in measles? So the genotyping sequence data inform us about importation, about transmission pathways, and how we are dealing with the elimination phase, with the documentation that we need to present, that we need to prove that our outbreaks and that we do not have more endemicity in our country. But as I told you before, we still have some limitations, as the little diversity in outbreak settings makes more difficult to interpret the cases. So we need to move to whole genome sequence? Probably yes, but another question is this information change the immediate public health response to a case or a sporadic case? So how many efforts we need to put on this? So at the moment, there is a working group at WHO working in this question, because in terms of measles, we were able to eliminate measles in the Americas, and Brazil and Venezuela lost it some years after. But the answer, or everything that we try to do in terms of immunization, in terms of epidemiological data and genomic data, were as simple as possible to have an impact in public health response. So we need to keep it in mind how to work with the data and which kind of data we extract for us to have really an impact in our public health response. So let's go to influenza. So why influenza is important? We all know that influenza can vary from year to year. So this genomic and antigenic analysis of influenza is very important to inform vaccine composition, to identify vaccine mismatch, and to track and to see the emergence of virus and strains. And all this is not only to inform the composition of the vaccine, but also to provide rapid information for local and national control measures. So influenza is a vast world. We have different animals that can have the virus, that can that we identify the virus. So it's much more than we can see here. If we do metagenomics, of course, we'll find the virus in many other species. So influenza is anywhere, is with us in the nature, in our day by day. So we need to use strategies that allow us to prevent, and genomic is one of these strategies that we can have good information to use control measures. So we can see here the influenza H1N1, the first pandemic of this century in 2009. And this influenza virus was a quite interesting virus, a triple resortant with genes from different species like H1N1 from avian virus, from classical swine, and the H3 from seasonal human influenza. And in some period, these different segments of influenza genome match, and we have a new virus that the human beings do not have yet antibodies or protection completely for this virus. So we have the pandemic. So WHO is working hard with their LabNet and epidemiologists, because it's necessary that each year we review the composition of the influenza vaccine. And what we are observing in the last 3 years, for example, in this table, that in the Southern Hemisphere, this composition do not match for all the influenza. For example, H1, H3, this Victoria has changed the strain in the last years. The Yamagata was more stable, is the same. So this is a big challenge for the surveillance. And it's a big challenge when we think about vaccine effectiveness. How can we use the tools that we have, the genomic tools, the epidemiological, classical epidemiological tools, to have a better answer for this? So we can compare here, for example, in the top, the single dose of measles vaccine that, independent of the genotype that is circulating, it gives us around 92% of seroconversion or protection in our population, just a single dose. But with influenza, we know that the vaccine that we have available now, or since the start influenza vaccine, is not our dream, is what we have. And so we have all the interferences of this changing in these drifts in the virus that can impact in the vaccine effectiveness. So how can we use in these two different questions that we need to answer for the measles program and for the influenza program, how can we use the genomic analysis to help us? So for measles, seems to be simple because the vaccine is really good. But for influenza, it's much more challengeable. So the use of different platform, genomic, antigenic, the immunization, we need to be careful how we use all this all together to answer the public health measures. So the influenza vaccines strain selection, the WHO has a global lab network that characterizes every year these antigenic drifts. So the analysis of the current strains, the antigenic and the genomic sequence is absolutely important for this definition. And we know that sometimes new strains always emerging, and sometimes after the choice of the vaccine is made. So we need to do a continuous work in this program to have a better understanding. So now let's go to SARS-CoV-2. So over the years, we can see the emergence or the detection of new viruses. Of course, the methodologies have been much better now for us to detect the new viruses. So let's start with influenza in 1933. So, and in the 50s, RSV, that is a big challenge annually for our children, and rhinoviruses in the 50s and 70s, 2 coronaviruses were identified and is here circulating with us. In the year 2000, or around the decade of 2000, we have the metapneumovirus, SARS-CoV in 2003, and 2 coronaviruses, one in 2004, another in 2005, and also Bocavirus in 2005. So, and MERS in 2012, and the long list, like Ebola, for example, that we have. So how can we go to this world and try to discover it, or try to be before all these viruses emerge and to know them better? So this is a quite interesting paper from a group in China showing the discovery using genomic data, genomic methodologies of a pool of SARS-CoV in bat related coronavirus. So if you go to the nature, we'll find the virus. So this animal and human genomic analysis must be continued doing, because we'll find the virus there, and we need to know this for us to prevent new emergence of virus. So for now, for example, we have for the SARS-CoV, we have the bat and we have the pangolin, and we still do not have in which animal we had the jump of the animal to human of the SARS-CoV-2. And this could happen. This can happen anywhere in the world. Can happen here in Rio de Janeiro, can happen in Switzerland, can happen in China, anywhere. In the way that the human beings are behaving with the nature that we all know that is not the best one, also is in favor that we can have this jump easier or quicker than we would like. So while we are doing this genomic surveillance of SARS-CoV-2, it's a new virus in the human population, and it's spread widely. So it's an RNA virus. So this can lead to change driven by virus adaptation to a new host. And this change can be in terms of transmissibility, as we are seeing now with the Delta variant, or pathogenicity, or both. And we need to have a good vaccine to prevent clinically severe infection. So, and what is the risk of the virus evading this vaccine protection? what is the risk of this? So WHO developed a nomenclature to add the communication between the population and the academia. So we have what we call variants of interest that are new strains that are emerging and have a potential of different potential in terms of given to spread more easily. And we have the variant of concern that we have sufficient evidence of increased transmissibility and or antigenic changing that can lead to decrease of vaccine effectiveness. So the genomic here helping directly the efforts and have an effect in policies on public health. So with the coronavirus, we have an excellent demonstration how we need to work together. So this group in Berlin and the Charité group, as soon as the Chinese released the sequence of the SARS-CoV in January, this group has a strong, they work a long time with coronavirus. So soon with the other researchers from Europe, they developed a real-time PCR that was critical in the beginning of the pandemic for all countries around the world to have access to the diagnose and to understand the epidemiological picture of each country or each region. So PAHO organized since the beginning a genomic network, and this genomic network is based that some laboratories of PAHO has can sequence samples, and this sequence they can receive samples from other countries. So is a support that the region is given one to other, exchange samples and exchange knowledge and improving the genomic in the region. So every country also organizes itself. So this is Brazil that Fiocruz together with the Minister of Health, and the Minister of Health at the beginning using, as many countries use, as WHO used, the national influenza laboratories to start the answer, and after to training other laboratories in the region, not only for real-time PCR, but also for genomic to improve the quality and the number of sequence that we need. So everybody started doing protocols for sequence of SARS-CoV-2 using different platforms. So Ian explained very well the nanopore and how useful it is. So we have also Illumina, Ion Torrent, Sanger, so different platforms has been used. And with this, we and all the other countries were able to observe and to demonstrate the introduction, the dispersion, and the evolution of SARS-CoV-2 in the beginning of the pandemic in each country that were able to have this. Another question that the genomic surveillance was very important is about how is reinfection? Because in the beginning, we don't know, will you have reinfection with SARS-CoV-2? This will be important, how we define reinfection case. So again, the genomic data is one of the key issues for the definition of reinfection cases. And the new variants of SARS-CoV-2 emerged all around the world. Are all these variants or all these mutations important? No, they are not. But we need to follow, we need to continue with this genomic platform for us to have a better understanding of the impact of SARS-CoV-2 in our country. So here is the emergence of the Gamma variant, the P1 in Brazil, that in the beginning, where it started in the Amazon, was a huge impact in morbidity and mortality. And we take the opportunity for this, and as other countries did, we develop a real-time PCR for variants of concern detection, was interference protocol that we can do like a screening. And here came the other question, which is this number that we use to say we have a good surveillance in our country, how many samples we are sequenced, and what this is telling us. So this magic number is very dependent of also the capacity, the financial support of each country to do this kind of investment. Of course, everybody to have the same thing as England, many sequenced by week, but this is not the reality. So we need to work better in this and try to continue, because the emergence of different variants, as we can see here in Brazil, the P1 with new variants, and that can have mutation that can be important for the vaccine, for the neutralization in the different vaccines. So also the genomic platform is permitting us to know better all this difference in genomes and to work with the classical virology like neutralization tests to understand the human sera of convalescence after infection, natural infection, and the human sera after vaccine, how is it in relation to neutralization for the different variants? So this vaccine effectiveness is absolutely important for control measures. And again, genomic is giving us this result. So this is a propaganda of our website. It's a fantastic one. I appreciate very much. So we invite you to see it. And we can find this website like temporal distribution of SARS-CoV-2 in different states and places in Brazil. So some points for us to end this discussion. So we need to work hard in each country to have a structured sampling frame and standardized protocol. This is very important. This genomic can give us an aspect or can show us the part of, for us to understand pathogenicity, transmissibility, host response, and novel mechanism for interventions. But how we will sustain this in terms of infrastructure is very challenging. So it requires continued financial support for this. But this financial support, if we continue with this, we can have an excellent opportunity to effectively implement genomics as a key part for future surveillance strategies that can inform public health policies. And so we will be better prepared for current and future pathogen threats that we will have. So I'd like to thank you very much for this opportunity. Again, I apologize for the beginning of my talk, and to thank some friends at Fiocruz, Juliana Leite and Pajo that helped me with this presentation, Pajo that we are always together, and the Fiocruz and GitSaid, that's this excellent platform for genomic data. Thank you very much.
Thank you so much, Marila. Actually, it was very nice, to have you back and for all your efforts actually to be back.
Y tiene toda la razón. Estamos aquí las dos aburridas como ostras.
I think, your message were very strong, and I think they are very much in line with what we would like to accomplish with this series of webinar. And I guess we will come back in time on the more in-depth discussion of the many important points that you raised at the end, the importance of the network, the importance to have as well consideration of the financial consideration, and as well the issue of standardization of methods across the different labs and countries. We have, I think we are over time, and so I guess we need to proceed to the closure of this first meeting. As I said, it's just one of the three of one. From my side, I would like to definitely thank all the participants, all the countries who participated, the panelists and the colleagues who made this webinar possible. And I'd like to turn it completely over to Jairo so that they can close the meeting as they better see fit. Thank you so much. Over.
Muchas gracias.
Thank you, Andrea. And to conclude, of course, I'd first like to thank our speakers once again, Dr. Cheney, Dr. Moshili, Dr. Moshili, and of course, our dear Dr. Marita Siqueira, who in a keynote way have given us a sample of all the informatics tools, which are the basis of molecular epidemiology, but above all the application in real scenarios as a response to public health surveillance with different examples and pathogens beyond the COVID-19, which is what we actually wanted to review today. Without a doubt, and as said by Dr. Andrea Vicari in his opening words, never like today have we been that interested in sequencing processes and its use in epidemiologic surveillance. Of course, due to the SARS-CoV-2 pandemic and an explosion of sequencing in the global databases and a speed which sometimes goes beyond our own ability to analyze. That's why molecular epidemiology gives us a chance to take all that information that we're generating in the countries and make some sense out of them, completing it in a much more efficient way with processes like the contact procedures and finding distribution patterns of distribution or circulation of the different pathogens, define conglomerates and changes in the epidemiology or clinic of the different agents that could be in circulation. And of course, for this, we need the raw material. And I'm talking about the sequences, not any simple sequence of a simple sample has to be used. They have to be high quality sequences as we saw today, and we see that we can control that. It must respond to clear selection criteria, allowing us to really respond to the surveillance questions we're asking. And that's why, as we've mentioned, and as Dr. Marilda said in her presentation, PAHO is working for over a year now in implementing and strengthening the regional sequencing and genomic surveillance network, which allows us to generate high quality sequencing and sharing information for better surveillance. And that's why our invitation to continue sequencing in the countries who have the possibility of doing so. We have countries with a lot of strengths and have been able to sequence and generate a large amount of information, but also those countries that have more limited capacity, sending samples to our reference laboratories through the network, and in that way ensure the continuity of the surveillance processes. And of course, to have greater information to study the molecular epidemiology. Please let us remember that this information we're generating in the laboratories must be orchestrated with all the departments and national surveillance systems in the country, because in that way we're going to be able to obtain the data that must be used in the decision-making processes. With all this, then, all I have to add is thank you again to our speakers and to all the participants and delegates from the different countries for taking their time in the midst of so much work to spend their morning with us. And those of you who stayed till the end, thank you very much. I'm sure some had to leave earlier because they had other things. Thank you to Dr. Cristina Valencia for the webinar, organizing it, and the coordination of Juliana Leite and the support of Dr. Leonel Kresh from the Laboratory Response Group. As usual, our thanks to Jessica, Maureen, Ronnie, who are working behind the screens, our translators who are so patient with us despite all the technical challenges, and to all the people who have been supporting us in one way or another in this first webinar. The presentations and recordings of the sessions will be available very soon in the Dropbox according to the emails recorded or registered, in our databases. And finally, remind you that our next session will be on September 28th, and we hope to have you all there We have 4 important speakers at that time from different institutions, including Sanger from Imperial College, University of Barcelona, and the College of London. The agenda will be available about 3 days before the seminar, so you will have access to it, and we hope you'll be with us there as well. Once again, a thank you to all of you for your being here, and we'll hope to see you again in the next webinars. Once again, thank you.